Welcome to this video presentation. During my PhD at EPFL in Lausanne, I specialized in developmental genomics and learned about the wonder of Hox genes that are the architect establishing body plans in animals. In particular, I defined how Hox genes can pattern the formation of the arm, the hands, and the wrist, that is the structure connecting both extremities. Later on, during my postdoc at the Max Planck Institute for Molecular Genetics in Berlin, I kept on working on the regulation of genes during the development of organs and embryonic structure, but this time with a specialization in human non-coding mutation. I have been involved in understanding how certain mutations lead to change in the 3D organization of the genome, inducing misexpression of genes at the wrong time or at the wrong place, and ultimately resulting in pathologies like skeletal malformations. Since December 2018, I opened my laboratory at the University of Geneva, where I work with talented scientists to understand the molecular processes that control gene expression during embryogenesis. These molecular processes are what define our body plan, the construction of our organs, and ultimately their function. Indeed, we humans have roughly the same set of genes as a fly, and differences between us and a fly mostly relies on how we use this gene. More specifically, we study how the genome, and in particular the chromatin, is instructing when and where a gene is activated during the construction of organs. Moreover, we define how mutation perturb these activities and ultimately induce malformation. To do that, we are using a broad range of methodologies. In particular, our laboratory relies on genetic engineering of mouse embryos using tetraploid complementation technique, which allow us to drastically reduce the number of animals used in research. To do this, we can count on the expert help of our faculty transgenic facility. We then use state-of-the-art technology, including high-C, ChIP-seq, or single-cell RNA sequencing, to characterize the factors that define gene activity in these engineered embryos. To efficiently implement this methodology, we can also count on the support of the IgE3 sequencing facility. Our multidisciplinary approach between functional genomics and developmental biology is linking what happens at the scale of the DNA and chromatin to what happens at the scale of morphogenesis. Our research will help us understand the fundamental genetic principles that drive morphogenesis and determine the mechanistic origin of many developmental malformations seen in the clinic.